Next up, we have Dr. Jen Gordon. Dr. Jen Gordon has been with the PURA Syndrome Foundation for probably 4 years now. Is that right? Have you? I know you 5 years, 4 years, for as long as we've been around that I can remember. So Jen is a research assistant professor at the Lewis Katz School of Medicine at Temple University. She is the research host, medical host here for this weekend, as well as 2 years ago in Philadelphia, where she very kindly opened up her research lab, which was an entire floor of a high-rise medical research facility in Philadelphia. It was quite impressive. But Dr. Gordon has provided many different things. She provides mice models. She and her team provide raw data. They also research other conditions. And so with, can I say that you, 2 years ago, she provided a short lecture that was non-medical, actually.
Oh, sure.
Jen has her own rare condition. And so it's personal on a different level. I mean, right? This is personal for all of us, and she understands it. And so she has always been very generous with her time and help here. So, Jen, thank you very much, first of all, and welcome, Dr. Jennifer Gordon.
So this is a challenge. I have to speak loudly. I can't see the pointer on the screen, but I'll do the best that I can. So if you can't hear me, do this, okay? So, or interrupt, and feel free to jump in with any questions as we go through. This is the first of the talks about the model systems. So as you heard a little while ago, we've been working on the mouse model. We've actually been studying, as Dirk described, the gene PURA and the protein per-alpha for many years. Before PURA syndrome, we even knew about this disorder. So I'm going to tell you a little bit about why we use the mouse, and I'm also going to tell you a little bit about the frog for our colleagues working on the frog from the UK that are not here. So in research, we use many different animal models. A couple of my favorites are the mouse and the rat, but I also do some primate research, and you can recognize some of these other ones here. Most of you have heard of Dolly, the sheep that was cloned many years ago, so that's a common model to be used. And Drosophila, certainly this picture is not drawn to scale because you can see this fly is huge compared to the other animals. But Drosophila, that's an animal model. It's a very powerful genetic model. And then we're going to hear a little bit about the zebrafish later on. So there are a number of advantages and disadvantages that Dirk highlighted. We're going to talk about all of the ones on this list except for primates today. And each one has its own little niche, and researchers are usually experts in one, maybe two, but not so many. So that's why we collaborate, because we can be the expert in one, but not everything. So we work together and share our findings, and this is actually gives us more powerful information, because if we find the same thing happening in multiple animal models, then we can all compare notes and get more information. So Dirk had mentioned that the mouse was very expensive, but that's all relative. So working with the mouse is more expensive than working with the fish or the frog, but it's a lot less expensive than working with primates or pigs or some of the large animals. And of course, working in human clinical trials is more expensive than any of the animal models, so it's all relative. So you're probably familiar with some of the steps involved in developing a drug. This is an example from cancer, so I just thought I'd walk through this. In the beginning, we often start in cells in the laboratory, and these cells may be from a patient with a tumor, or normal cells from a patient, or from a patient with a genetic mutation like PURA syndrome. And then we can study those cells in culture, see how a treatment affects those cells. And then usually the second stage is to use an animal model. So if you want to be able to get approval to use your drug in a person, you have to show that it's working in a model. And mice are a very common model because they're easy to use. And then the next thing is to, and that's to make sure that what you're doing is safe. okay? So the first purpose is to do no harm and make sure that what you're treating the patient with is not going to hurt them, right? So we do a safety study, and then we do that in the animals. And then we also want to see whether it works to treat the actual disease. So we create a model in an animal that will recapitulate the phenotype of the disease, the different symptoms, the different features of the disease, and we would hope that whatever treatment we're using is going to change how the person is behaving. And then that would get submitted in the US to the FDA and get approval to use that drug, and then you would try it in a clinical trial, and then if all goes well there, you go through a couple different phases of the clinical trial, and then hopefully the end result is that you have an approved drug for that particular treatment, for that particular disease. So how does all that happen? Well, in PURA syndrome, we're talking about a developmental disorder. So if we want to use these animal models, the first thing we have to do is to pick the right one that's going to give us information about the development. So here's an image that shows you many different species. They're all listed at the top. Fish, turtle, chicken, pig, and mouse. And you can see at the top the single-cell embryo. So you know you have an embryo that gets fertilized, and then cell division begins. And then gradually you start to form the developing embryo. And if you take a look and you see these early images, early in development, they all look remarkably similar because there's a similar program that vertebrates and eukaryotes use, and the way that the cells are programmed to develop the organs throughout the body, etc. So there's a lot of similarity between these models. And that brings us to the frog. So this is Xenopus, or the frog. And I have a few slides here, courtesy of the Xenopus folks in the UK, about what is Xenopus. So there's a couple different species. There's Levelis and Tropicalis are shown here. You can see that the larger frog is the female, and the smaller frog is the male of the species. So they get bred together, and they create embryos, and then we can study the development of those embryos. So frogs are really fascinating creatures. They communicate using sound, not necessarily visually, and they're also carnivores. Most people don't know that. And they're native through most of sub-Saharan Africa, and now they're grown in research laboratories all over the world in these little captive environments where we can control how they breed and multiply. So frogs have been studied for a very long time. They've been used in the clinic for a number of years for different purposes, because you can put them in an environment with water and other buffers and be able to put chemicals in the water and expose the animals and then watch and see how they respond, because they absorb everything through their skin. So it's a very effective way to deliver a drug or a compound to the animal and study its response. So this is the life cycle of the frog. And one of the really cool things is if you see how fast they develop, right? So you can see here, starting over on the right, the single cell stage. So you have fertilization, and then that single cell becomes 2 cells, becomes 4 cells, et cetera, et cetera. And as it begins to develop, you can see that it starts to create what's called a neural tube. I can't find my pointer here. There it is. It starts to form this neural tube. This is the beginning of the nervous system, the spinal cord, and then all the cells kind of already know what to do, where to go. They have a polarity, they have an organization. And then if you look at the number of hours that it takes, you can see that it's only about a day and a half before it becomes a tadpole. So you think about the researchers in the lab probably can't sleep because they have to be there to examine every single minute, because so much is happening so quickly. And you can go on YouTube and you can find videos, time-lapse videos of this development. It's really cool. So I encourage you to take a look. And you can catch all of those individual steps that are happening. And then there's about a 12-month cycle until it becomes an adult. And that means also that you can breed the animals every few months, create new embryos in the lab whenever you need to. So it's a very quick model. So these are some slides from the Xenopus Research Resource Center, the EXR Sea, in Portsmouth, UK, where Dr. Gilly and his student Annie are located. So you can see their pictures down at the bottom. So you can see in these images the aquatic setup where they keep the frogs. So some of the pictures on the right, you can see the research laboratory where they do the pipetting and the analysis of all the materials. And then in the center column, you see those big areas. Each of those little containers is where the frogs are living, and they can separate the moms and the dads and then put them together when they want to breed them. And then the tiny tadpoles that are coming out, and they control each of those tanks. So they're in an environment where we know what they're eating, what kind of water they have, what temperature it is. All the conditions are very closely monitored. So they're very, very well taken care of. We saw this slide a little bit earlier in a different form, showing you how rapidly everything develops. So what the folks in the UK are doing are they're looking at the PURA gene because, as you heard Helen mention, this is a very highly conserved gene. So you can find the PURA gene in bacteria, in frogs, in worms, in mice, in monkeys, in humans. And this tells us it's a very old and very important gene because it hasn't changed. Because if it changed, then the organism, we assume, wouldn't survive because it's so important. So what they're doing in the frog is to label this protein so they can see what happens when they make a change in the gene that's going to affect the protein, similar to what you see in PURA syndrome. So the first step is to remove the gene. So these are some preliminary results from their laboratory, and you can see here is the head of the normal tadpole and what shape it usually has, and you see the eyes on both sides of the head, and then you can see next to it there are two examples of animals that are lacking the PURA gene, so they're not making any per-alpha protein, and you can see that the shape is very different. So we know automatically that there are some differences between the normal and the PURA syndrome frog. And they've so far seen structural abnormalities in the way that the brain is developing. They can see the way that the tadpoles are swimming is a little bit different. And so we know that kids with PURA syndrome have some movement abnormalities. So it looks like this is seen in the frog as well. They said they have some feeding difficulties with the frogs, which again is common to PURA syndrome. And you can see the shape of the head and that PURA face that Margot has described. So this is sort of recapitulating that. And they also have some evidence that the frogs may have seizures. So this could be a good model to try to look and screen for anti-seizure medications. And that's one of the priorities that we hope to work together on. So I'm going to move on to the mouse, but does anybody have any comments about the frog? Anybody have any pet frogs at home? Be nice to your frogs. They're very useful for us. Okay. So we'll go on and talk about the mouse. Yeah. Yes. Yeah, so the first experiment they did was to cut out the whole gene, right? Correct.
Did everyone hear the question?
Yeah, I'll repeat the question. The question was the PURA affected tadpole, was that removal of the whole gene or a mutation or something like that? So the answer is that it was removal of the whole gene and both copies, right? So we heard from Helen and we heard from Dirk, we have 2 copies of the PURA gene, and if we remove 1 copy, this would be like a PURA patient with a deletion or a stop codon in the very beginning of the protein. If we remove both copies, we don't know of any patients that have both copies removed, We've done this in the mouse, and I can show you that those mice don't survive, so we don't think that there are any patients out there that would have both copies deleted, but we don't know. So we have, in PURA syndrome, one normal copy and one mutated or deleted copy. And that's what we're trying to show in the different animal models. So in this example, they've removed both copies, and they see very dramatic changes. Their next step is going to be to remove one copy. So they have one normal and one mutated copy. Then the next step after that is going to be to put in some of the PURA syndrome mutations that we see in the children to see whether or not they look the same. Good question. Yeah.
So that means that frogs can live with the removal of growth.
Now, I'm not the frog person, but I can see that this tadpole is surviving at this stage. What happens to it when it becomes an adult or later on toward adulthood? I don't know the answer to that yet. Yeah. But they survive through this stage. Our mice survive till about 3 weeks of age, which is maybe early teens that are missing both genes, and I'm going to show you that in a few minutes. Other questions, comments? Yeah. Yes, they're doing it at, the question is, are they doing it at the single cell stage? So would every single cell in the tadpole be missing the PURA gene? And that's the first experiment, yes. There are more sophisticated models that we can do where we can figure out how to turn or turn off the gene at different stages during development or in certain subset of cells. But this is the first pass of the experiment. So this is very encouraging because they can see a phenotype. They can see a physical change, and they can see some of the behavior changes. So this is very exciting in the lab, and very encouraging to do more experiments like this. You mean in the animal, or how long the research takes? Well, research, I would say this is a graduate student who's been doing this project for about 6 to 9 months. Now, this is a laboratory that already knows how to do this for many genes. So they come up with their next favorite gene of interest that they want to ask the question, and they have a resource. They have a core facility. They have everything in place and all the expertise. And the head of the lab will talk to the student, design a new project, but that student already knows how to do many of those things, so they're not starting from scratch. These are experts. Good questions. Any more? Okay. So mus musculus, or the house mouse, right? Everybody's had a house mouse at one time or another, whether you wanted one or not. So this is where we keep the mice in the research lab. So they live in little condos that are all stacked together. And each one of those little boxes, you see the guy carrying the micro-isolator. It's got a HEPA filter on it. We take extreme care of these guys. I know in my colony I have hundreds and hundreds and hundreds of mice. I know each one of them. They each have a unique number. I have about 7 or 8 different lines of mice with different genes. One group of them are the PURA mice. We breed the males and females. We talk about them by their numbers. We get to know them, say, oh, how's 352 doing today? And the tech will be like, oh, he's looking okay, so we get to know them very well. And then when their experiment is done, we can collect all the tissues, and then we go to the laboratory and we're able to analyze all that material. And that's where I'm going to show you some of the pictures that we have. So it's a huge endeavor, very, very expensive and time-consuming. It can take 2 to 3 years to develop a new line of mice, and then you have to do all the analysis to describe them, and hundreds of thousands of dollars to do that. But researchers all over the world are doing that, and we share our mouse models with each other. So where do we actually get our mice? I know this is kind of seems strange, but there's companies and core facilities that trade mice with each other. So I can get online and order mice from a catalog, and I can decide, I want this kind that has this specific, it's really, really specific, right? It's not quite Amazon, but almost like that. And there are tens of thousands of different strains of mice that are available throughout the world. One of our most popular places is the Jackson Labs in Bar Harbor, Maine, not far from here. And you can go online and order extremely, extremely specific genetics, order different lines of mice, cross them with each other, and things like that. So it's really cool. This is a poster of 3 of the most popular strains. So you can see what they look like from pups all the way up to adults. So the little tiny ones that are off toward the left, they look like little gummy bears when they're born. They're really adorable. The first week, they're really cute. Then they start to get fur, and you can kind of stage them, see what they look like all throughout development. And they come in different coat colors. Sometimes we use that when we do our breeding for experiments. And then I wanted to show you this picture just because it's a really cool picture. So you can see on the left, this is fluorescent protein. So we've added this fluorescent gene it's a gene that makes a fluorescent protein. So when you turn out the lights, the animals with the gene glow in the dark. So it's really neat. Now once they get, you can see which ones are glow in the dark, right? There's 2 on the left and then 1 on the right and then 3 in the middle that don't have the gene. Right, so when we breed them, it's inheritance, just like with inheriting any gene from mom or dad. What's the possibility that you're going to inherit it or not? So we do all that kind of genetics in the lab. And then the other thing we can do is to label specific proteins. So the panel on the right is a label for a protein only found in neurons. So those little structures that you see are individual neurons. They look like a little bulb. That's the cell body. And then you can see all the branches of the tree coming out, and that's where the neuron is reaching out, and it's going to connect to other neurons. So where does that gene come from? It comes from another animal, the jellyfish. You've ever been in the aquarium, you see those beautiful jellyfish. Well, some researcher came up with the idea to figure out what that gene was, and now we use it in the lab all the time because we can label all the proteins that we want. It's really cool. Anybody have any comments or questions so far? Okay, we'll keep going. So when we first made the mice, this was going back over 15 years ago that we published this. So as I said, we've been studying per-alpha for a long time. We made our own gene. So we figured out how to remove a portion of the PURA gene and replace it with a marker in the mice so that we could see who had the gene and who didn't. And what we found down here at the bottom is that the mice that are lacking the gene, they don't develop properly, and eventually they will die compared to the normal mice. They don't gain as much weight, and they don't thrive, and they have difficulty moving and many different features. And then eventually, around 3 weeks of age, they won't survive anymore. And we did an analysis of tissue from the brain. This is a Western blot. So we took the brains of the mice, and we made protein, and we ran it on a gel, and then we probed it with a detection for per-alpha. So this is actual protein per-alpha, and you can see the wild type is in the first lane, mice with 1 copy of normal per-alpha and 1 missing in the middle, and then in the third lane is no copies of per-alpha. So there's no per-alpha made when the PURA gene is gone. But the mice in the middle that have 1 normal copy and 1 deleted copy, like PURA syndrome, have less protein. So you see the intensity of the band in the middle is different than the band on the left. And that was the first sign that there might be haploinsufficiency, the word you heard Helen talking about earlier, which means that 1 copy, 1 normal copy, is not enough to make all the protein that you need in the brain. And this is one of the main things that we're trying to understand. What's the mechanism behind that? Why is it that 1 copy is not enough? And what can we do with the 1 copy that's there? Can we find a way to increase the amount of protein being made from the existing copy? Yeah. 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 So actually, when I was my first job in the lab, 28 years ago, I was working on this protein because we were looking for a gene that was involved in myelination. And this particular gene we found was important in developing brain. So we wanted to figure out exactly what it did. So we knocked it out in the mouse so that we could study what the effect was. And at that time, the mouse genome had not been sequenced yet, so we actually had to clone the gene and figure out all the details about how it worked. So this is basic research because we thought it was interesting. Yeah. And that's a really important point because most of the work that the researchers were talking about here hasn't, as Dominic mentioned, is not funded by the foundation. It's funded by many of the government agencies. This is basic research. These are taxpayer dollars at work. Most of my funding over the years has come from the NIH. Supports students in the lab, supports core facilities, and this is the reason that we've been able to make so much progress, I think, in PURA syndrome is because of all the background work and the training that many of the researchers have had over the years, and now we can apply our resources specifically toward understanding how it applies to PURA syndrome. Other questions? Okay. So this is when we first discovered that PURA was expressed in neurons. So I showed you those green neurons that had the bulb and the tree coming up. Well, this is what the sections look like in the brain. So we take the brains from the mice and we slice them up very thinly on the microtome into these little tissue-like pieces that we can then mount on a slide and look under the microscope. And we can stain them with dyes and chemicals, and we can stain them with antibodies that detect the protein. So you can see this region here, the brown staining, is the PURA, and the blue in the middle, those round-like cells, that's the nucleus of the cell. That's where the DNA is found. And you can see the brown staining around it, and the cytoplasm is the PURA. And this was the first clue that we had that PURA was so important to neurons. So we've been able to do a lot of studies where we've found that PURA is important for developing neurons. And if you don't have PURA, you don't get enough neurons. And the neurons that are there, they don't have as elaborate processes. Their dendrites that we call them, the branches of the tree, don't develop as well as they do when the PURA is present. So we're trying to understand what the impact is, what are the mechanisms at the cellular level that PURA is controlling that helps to neurons to grow and to survive. And we think that's really critical to understanding what the defect may be in PURA syndrome, in the brain. And just here are some more pictures. On the top row are all the normal mice, and on the bottom row are the mice lacking PURA. And again, you can see those neurons with the brown staining, and on the top row, how elaborate those structures are, like the tree, but that the neurons on the bottom do not have that. And of course, it's much more complicated than that. We know that the proteins move around during development. This is an example of PURA in the cerebellum. This is the cerebellum is the area of the brain toward the back of the brain that's involved in balance. We know that kids with PURA syndrome have problems with gait and balance, and I'll show you what happens to our mice. And then another area, which is the hippocampus. This is an area that's really important in learning and memory. It's important in all of us. It's an area that gets affected a lot in older individuals with Alzheimer's for cognition and learning and memory. So many of the areas that are important in PURA syndrome are also critical for all of us at all stages of life. We're going to hear about cell culture models in human cells, but we can also culture cells from mice and rats and study them and learn a lot about the details outside of all of the other cells in the brain or the animal and study them specifically in the dish. And then we can also look at the molecular mechanisms. And this is what we spend a lot of our time talking about around the table yesterday and back in the laboratory. And what exactly are these molecular pathways and cellular pathways? And how do the proteins talk to each other? And what's regulating what? And what happens when PURA is not there? Is there another protein that's taking over? And Dirk was mentioning the cellular friends of PURA. If PURA is missing, what happens to all of its companions and the other proteins that are interacting? And that's what we spend a lot of our time doing. So we're interested in looking at the behavior of the mice to see whether they behave the way that we would predict children with PURA syndrome tell us they should. So mice are kind of neat. You can do lots of behavioral stuff with them. Some of the studies are very sophisticated. This is a setup. You can see a camera with a box. We put the mice in the box and we watch them explore. Mice are very curious, and if they're comfortable, they'll kind of walk around and see what's going on. And we have computers that track their activity so we can see how much time they're spending. Mice like to run to corners and not be in the middle if there's really bright light. You know, they kind of seek the shadows and they want to get out of the way. So we can see whether they behave normally. And then we record all that information, and the lucky graduate students get to look at the videotapes and score all the results and see if they can find a difference between our control and our PURA syndrome mice. And here's a picture on the right of one of the mice in the apparatus going through the experiment. Some of our experiments are not as sophisticated, a kind of old school approach. This is one way that we determined there were gait changes. So we took the mouse and we dipped its right paws in one color ink, and we dipped its left paws in another color ink, and we put it on some butcher paper and we let it run, right? Not easy. The mice don't really like that very much. So you have to have a mouse that's cooperative. Not all of them. Some of them might just sit there and be like, no, I'm not going to do what you want me to do today. So other mice might be more, friendly, cooperative, or in a better mood, etc. So you can see the changes between the images on the left and the right compared to each other, the wild type versus the PURA syndrome, where the PURA syndrome mice are kind of dragging their feet and the way that their gait works. And I can tell, if I'm looking in the cage, I can tell a PURA mouse from a wild type mouse because he's got a little bit of a waddle, kind of sits back a little bit, and he doesn't walk quite the same way. So some of the techs think I'm crazy, but I'm right, because I'll point them out and they're like, that's it, that's you can see it. So I'm sort of a mouse neurologist, right? I've gotten to know these things. I don't know anything about human neurology, but I can tell in the mice. So what does this mean? This means that there's a deficiency in the cerebellum, which is the area that controls balance, and I showed you some of the images. So we have a way to measure in the lab if there's a change in the cerebellum, and this also means that if we can find a treatment that we think may affect the cerebellum, we have a way to measure if it's working. So we can treat the mouse and then we can go back and measure its gait. So this is an open field test. There's two of them that are very popular. One's called novel object recognition. The other is novel object location. I guess I refer to it as one of these things is not like the other from Sesame Street, right? So we put a bunch of objects in the box, and we put the mouse in the box, and the mouse goes around and looks at everything. And then we take the mouse out. We do that a few times. And then we wait a few days or a few hours, and we put the mouse back in, but now we've moved something. Now, if the mouse is able to remember what was there before, he's going to see what's new, and he's going to run over to what's new and say, hey, I don't remember that. That's interesting. What's going on? This is something new. Just like people, they want something new. You go to the buffet, you want to eat something different, not what you just ate, right? So we tell the mouse, we ask him, do you recognize that something's different? And that's how we can see whether or not his hippocampus, which is the area of the brain that's important for that, is working properly. And this is how the data looks when we analyze it. On the left, you can see the dark square is where the objects are. So we go and we do these trials and we move the objects around, and then we see whether the animal remembers what we moved and where we moved. And then we can graph those results on the right. And the bottom line is it shows that the PURA syndrome mice have a problem with the spatial recognition. So they don't remember when we move the objects around, and they don't remember when we change the objects. And that makes sense because I showed you the hippocampus, and I showed you that the neurons are not the same in the hippocampus, and it's the job of those neurons to remember new things. So when you go and you're learning things, your brain is always learning and making new memories and forming new things, the hippocampus is one of the areas that does that. So your neurons are always busy making new memories and laying down those new thoughts. Yeah, we can. So the question is how much time do we wait between the testing? Do we wait a few hours, a few days, a few weeks, a few months? And it depends on the question. If we're studying something like neurodegeneration and Alzheimer's, then we start with an older mouse and we wait and we see what happens over time. If we're looking for an immediate change and we don't have to wait for neurons to change, then we may test within hours. So there are very different ways to ask the question. The same way when you go to the doctor, when they do a test for baseline or they do an immediate test and see if you've had any improvement, they may not see something until 6 months or a year later because it takes time for that to progress. So it depends on what question you want to ask. Yeah, we have. I'm going to show you something right now. Good question. So this is the Barnes maze, and this is there's this big spiral and this wheel, and then each of the wheels, you see the little holes around the outside perimeter. Each of those is a potential escape hole, but they're all fake holes with just a false bottom except for one. And there's one hole that's got a big gap underneath, and the mouse can run in and hide. So we put a big bright light and noise, and then we drop him in the middle and we say go, right? And he's got to find the escape hole. And then we put little symbols, so you see the plus sign and the square, and these are visual cues, and we can use scents because mice respond to scent and smell very much more than we do. And this is kind of like the where did I park my car? That's the way I think about it, right? So you come out of the airport after the meeting, and you got to remember what level am I and where did I put my car? All right, so this is what we do to the mice. And the mice are really clever. Sometimes they try to cheat and look over the side of the wheel and see where the escape hole is. So you have to really keep an eye on them so they don't try to cheat. So this maps again to the hippocampus. And this is a test that we actually study over many weeks. So we do a bunch of trials and we test them and we teach them and they learn where the hole is. And then we move the hole. And then we see, do they keep going back to where the hole used to be, and how long does it take them to catch on that it's not there anymore? And we can also look and see what's their search strategy. Do they look in every hole in a row? Do they randomly look at some or the other? And it's pretty cool because some of the mice will just kind of go through every single hole very patiently, and then others will kind of like randomly look around, and we see what does that mean. And in our PURA syndrome mice, we've been able to show that they do not remember where the hole is like wild-type mice. Again, that's mapping to the hippocampus. And just a reminder, these are the areas, the hippocampus in the upper slides and the cerebellum in the lower slides. So the hippocampus being the learning and memory, and the cerebellum being the gait and the motor function. We're also working on some tests for peripheral neuropathy and pain because we understand that there's some question whether the PURA syndrome kids have normal response to pain and pain receptors in the periphery. So we can do this in mice. We put the mice on a hot plate or a cold plate and we change the temperature gradually, and then we see when they sense that it's too warm in here or it's too cold, and they move to another plate. So we don't make them stay there. We always give them an escape. So it's not a noxious stimuli. It's a very gentle stimuli, and they can always move if they're uncomfortable. And we see at what temperature they're going to move. And we have some preliminary results that shows that the PURA syndrome mice may not respond the same way that wild-type mice do. And again, this is a good way to measure because it's not as user variable. So when some of these other tests that we run, like I told you how the mice cheat, one of the technicians in the lab, Regina, the mice love her. So when she puts them in the Barnes maze, they run to her instead of the hole. So that can be a problem when you're trying to analyze the data. This one, we put them in the device, and the device does all the work, and we don't handle them at all. So it's much easier to run with many animals, and it's more objective. So I just want to finish up here to tell you. Oh, question? Yeah. Yeah, that's a good question. So so the question was, is it because that the animals don't think about moving, or that they don't sense the heat or the cold, right? And they're two different. So you have different pain receptors for heat and cold, and they're in the skin, and they operate separately. So the first question is, are the cold receptors affected, or the heat receptors, or both? And then many of these, like a reflex, when you touch the hot stove, you don't have to think about that. That goes to your spinal cord, comes back down, doesn't even get to your brain until 2 seconds later. You're like, boy, that was hot, but your hand already did all the work, right? So when we do this measurement, the mouse is going to move automatically, assuming it can move. If we had a model where it wasn't able to walk, that might affect the result. But these guys can move around enough that we don't think that's a problem, and it's a very gradual change in the temperature. So we think it is a receptor in the periphery. And we've been studying the brain, but we want to understand in the peripheral nervous system, and this could also be one of the questions about what is the cause of the hypotonia. Is it muscle contraction? Is it neuropathic? Is it something about the peripheral nervous system that we want to dive deeper into? Good question. More questions? Okay, so we are going to hear about the patient registry, and we know about biobanking. Yeah. So the question is about sound? Yeah, so when we originally characterized the mice, we did some tests to make sure that they respond to sound and loud sounds, because we want to make sure when we do the behavioral studies, if they can't see, for example, and we're giving the visual cues, we don't want to measure that they can't see. We want to measure that they don't remember, right? Because the test is about memory. Well, if we're giving them sound as a stimulus, we want to make sure that the reason that one group performs better than another is not because of a difference in sound, because that's not the test. So we go through and we try to measure all of those things. We haven't seen anything specifically affecting sound, but I can tell you that mice have much more developed sound than humans do, and they rely on it much more. So it may be that not all of the organs that we study are going to be exactly the same between human and mouse. Other questions? So we're working on our own biobank of mouse samples. You're going to hear later from Andrew about some RNA work based on the mice, and from Ulrich about metabolomics. I'm not going to say anything else, just to mention that we can take tissues from the mice that we can't get from the PURA syndrome patients. We can go in and collect samples longitudinally from the brain. We can collect blood samples in parallel, so we can see if we have blood samples from PURA syndrome kids, would that be the similar thing that we see in the mouse? So it's a very powerful way to use these samples that we can hopefully work on some biomarker discovery, and we'll hear about that this afternoon. And I just want to acknowledge there's a lot of people that have been involved in the work. This is not just me. My colleagues and I have been working on this project for 20-some years. And as I mentioned earlier, most of our funding is coming from the federal government for other projects that are related to PURA alpha, but not directly PURA syndrome, though we are applying for funding for PURA syndrome studies. And I want to mention Brittany. She's here today. She presented some of her work yesterday on immunohistochemistry in the mouse brain, so please find her and talk to her if you have any other questions, or me. We'll be here the rest of the meeting. So thank you for your attention.